just so you get more security with your scores. And I added to your spreadsheet calendar for February and March.
Did you have students in the past that pushed their exam twice?
Yeah, happens all the time. People can often push it back all the way to like, May if they want to, get their score back in time for applications, but sometimes that doesn't work, so they study it over the summer. So, yeah, it's a super common thing. So, yes.
Here's the thing. My only problem pushing it is I don't like that fact that like I'm just comfortable with the fact that like, oh, okay, well, I didn't get what I wanted. Like, yeah, I'll just keep pushing it on, you know? Like I don't want that and I don't like it. And also it's my last semester of college, I was looking forward to actually enjoying the whole semester with my friends and stuff because I've really just been a hermit. Um, but if I can take it in March still, that would be fine because I at least have a month
Yeah.
to enjoy my last weeks of college.
Yep. Yep.
Okay. Um, now, so you're thinking the 8th. Okay, I was thinking the 21st.
Oh yeah, no, this is just what was there from a different student that
Got it.
copied this from, but you said 21st?
So yeah, there's only the 8th and the 21st. I have break from the 9th to the 15th.
Uh-huh.
So, I mean, and then like, yeah, that would be like a good time. 21st would be a good time because I have break. My only issue too is there are no spots available right now. Hopefully they're gonna open up and I get those notifications. You know what I'm talking about?
Yep.
So have your students in the past like they were fine? Spots eventually opened up?
Hmm.
Okay.
So let's see. Um, oh, like November went all the way.
Sorry, yeah, I pushed it because I'm starting to get so many tabs.
Yeah,
Yeah.
I'm just trying to see like what we have because I guess this is where we scheduled like the AMC full lengths and stuff. So I'm gonna go backwards from the 21st. So
Oh, okay.
um let me see. Okay, so that's AMC full length 5, 4, 3, and
Yeah.
a lot of Altius ones. So okay.
Yeah. Okay, by the way, I might take these exams on Fridays then if my test is gonna be on a Friday.
Okay.
So 'cause I don't have class on Fridays, so like I feel like that would be the right thing probably to do.
Okay. So it seems like we're gonna end up with having you take uh one every week anyway, because that way we can get through all of the Altius
Got it.
links. Um, well, I have it so that you have like an extra week, I guess. Um, but just going backwards, like means that, you could take your Altius full length to like next weekend if you want, or this one, but yeah, this
Wait,
way.
repeat that. I'm sorry. I was zoned out.
Oh yeah. So basically I went backwards from your test date
Hmm.
a week before being the AMC tests, going backwards, and then going backwards with the Altius full lengths. So you have
Hmm.
basically like an extra week here. So you could take your Altius full length to like either this weekend if you like, or next weekend. But this way we're gonna have you finish up all 10 Altius full lengths, and then you'll do your three remaining AMC full lengths. And yeah, that'll be more than enough.
Okay, here's the thing though. Um, I know you're a big supporter of take as many full as you can, Navy SEAL training and all that.
Yep.
Um, the only thing with me with in full lengths, I just unfortunately I don't know. I'm just not built different, but I it takes me 2 days to fully review a full length. Like because the way I reviewed like the last LTS exam, like I literally picked apart like everything. And it took me I timed it, it took me like over 15 hours.
Mhm.
Um, so I would have to be reviewing these full lengths 2 days. I take them one full day and I review them 2 days. So it would be like 1/3 of my time is, taking full lengths and reviewing them. You know what I mean? It would be like three out of, and
Yeah.
I'm the thing is, I have school too, so I'm not going to be studying Mondays and Wednesdays.
Hmm.
Like
Yeah.
I'm only going to have 2 days then
Yeah.
for anything else.
Hmm. Yeah. So I guess then it's a question of, is there something that you would do otherwise that would be more important than reviewing the tests? I think that doing the tests, reviewing the tests would be like the most important thing to do. So it's okay if it takes like, I guess, 2 days to review a test. Um, because that basically covers everything. I mean, like, what else could you do, right? Um, like on days when you're not taking a test or not reviewing a test, you can do like, practice passages and stuff, but you're gonna get most of your work done from doing the practice tests and reviewing the practice tests anyway.
Okay.
So yeah.
Okay. So, all right. Well, I guess I'm gonna probably take Altius next Friday,
Okay.
then spend like the next week and a half just going over content gaps. So you recommend those TPR books, right? Or no, Berkeley, it's TBR,
Oh,
Berkeley Review
or
for physics and chemistry.
uh-huh. um yeah, if you find that they're useful in that, then by all means. But uh yeah, I guess we can talk about like what would be. So okay, when you review your Altius or whatever, when you review your full lengths, for like physics and chemistry, what do you typically use? I guess like you typically use like videos and stuff. So, it's whatever you find to be like whatever works for you in terms of like, if you try to read like some of the Berkeley Review book and it's like it's too much or whatever, then we can have you use like a video or something like that. Um, but yeah, I would say it's up to you, like whatever you find to be most whatever works for you in terms of like how you learn.
Mhm. I agree with you though that MCAT's a reading test, and I've been trying to like read more in terms of well just read, but also like to review stuff. Like I would read. I read the Saladin book a bit on like other stuff and
Yep.
um yeah, I mean it's just I think there's certain content gaps that I have that I'm just
Uh-huh.
maybe refusing to address, and I'm thinking that I just doing more like practice is gonna solve them. And with CP on this section, I got a 129. I never even got a 128. I got a 129. So I was like, okay. It was that the hit of dopamine was nice. Um, but I once again, if I'm being a realist, I think that this FL2 it hit some of my like good like things I'm good at, if that makes sense. So um I also just don't want to get obviously overly cocky because you know that I think there's certain things like torque and stuff that I'm probably gonna have to go into Berkeley and review.
Uh-huh. Yep.
But
Use that integrated MCAT website with the physics cards.
I have it saved. I have a bookmark. But like
That could be a an easy like first place to
okay.
at, like even before Berkeley Review, because it's more concise. So that could be the your first resource that you look at. Yeah.
That's right, pre-med village. Okay.
Yeah, the
Got it.
cards in particular,
The what?
physics cards.
I didn't even see that. I don't think I saw the physics cards.
Here, try this link.
Oh wow. Okay. Solid.
Yeah, so if you click on the flashcard chapters, it'll show you the different ones like fluid mechanics, waves, etc. So this could be the first thing that you look at when you evaluate physics stuff.
Yeah, okay. All right. Give me 1 second, I'll be right back and we get into the passage for the day. Okay. I have a question. When you like fully when you improved your psych soc like a lot, because I know that was like your weaker suit before, like I know you're really good from the jump at like CP and BB. How'd you improve psych?
Um, every psych thing that I did, I would make like cards to like, even if I got a question wrong, anything in the choices or whatever, if I didn't know what a term meant, I would just make Anki cards for it. And so I'll just learn, like, it just would learn as much as I can after each practice test and
Okay.
went in that direction. Yeah.
All right. Okay.
Okay.
So the transcription factor FOXP3 is encoded by an X-linked gene in mice and humans. Um,
What can you tell me about X-linked genes?
well, X-linked genes are both humans and um, sorry, X- X-linked genes are in both females and males. It's like a sex-linked sort of thing.
Okay.
Um, And males have X and Y, while females have X and X.
All right. So if it's an X-linked recessive, um, a female would need to be like homozygous in order to have that trait, right?
Yeah, so yeah.
But the male, since the male only has one X chromosome, if the male receives just one of that, uh, you know, um, recessive allele, they don't have another one. They don't have the ability to have another X chromosome to turn them into carriers. So if they just have one of that X-linked gene, then they exhibit that disease or whatever it's related to.
Yeah, exactly.
Um, what's a transcription factor?
So transcription factor, basically the way I understand it is every single one of our cells has the same genome, including promoters and enhancers.
Okay.
Now, different cells have different transcription factors,
Okay.
and that's why they um do different things. Different cells do different things because they have different transcription factors.
What does a transcription factor do?
From what I understand, it binds
Hmm.
the promoter
Yep.
and transcribes the gene to create mRNA.
Yep. Good. Okay.
Um, okay. Also, what is the operator? That's after the promoter, like
The
in general.
operator?
Yeah.
You see that?
No, 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 like in general, in general. You know how there's promoter, operator.
Okay. Wait.
Is that in prokaryotes only?
Yeah, I think that's related to the operon.
Yep, yep, yep.
So that would be in prokaryotes and viruses.
Got it. Okay. Approximately 60% of female FOXP3SF slash plus mice, which carry one inactivating scirphin allele and one wild type allele of the FOXP3 gene, spontaneously develop mammary tumors by 2 years of age. So, okay, female FOX3P, FOXP3SF. Yeah, so one inactivating, one wild type. Okay.
Mhm.
They've developed, it's X-linked. Okay, so it means that it's a dominant.
Um, let's see. They talk about this a scurfin allele being inactivating and the wild type plus allele of the FOXP gene, and then it causes this type of tumor to be grown. So perhaps it could mean that. Um, I don't know yet if it's recessive or not. Um, I guess it would be dominant then. Yeah, if you have one, if you have that one allele for it, you would get the tumors. But
Okay. The yeah, the following experimental observations clarify the relationship between FOXP3 and development of mammary tumors. Wild type FOXP3 transcripts are easily detected in non-cancerous mammary epithelium from female FOXP3SF plus mice, but are undetectable in cancerous mammary epithelium from the same mice. Um,
okay, so I guess it's saying that the wild type FOXP3 transcripts will be present in non-cancerous cells and the, you know, female FOXP3 SF slash plus mice, but are not in cancerous mammary epithelium.
Right.
So, okay.
In female FOXP3 SF plus mice, the wild type FOXP3 allele is carried on the inactivated X chromosome in 15% of non-cancerous mammary epithelial cells, but in 10% of cancerous mammary epithelial cells. Um, okay.
The inactivated X chromosome is what carries that wild type FOXP3 allele in, so that's located on the inactivated X chromosome in 15% of non-cancerous mammary epithelial cells, but in 100% of the cancerous mammary epithelial cells.
I'm getting a little confused. Hold on. So, because 60% carry inactivating scurfin, one wild type allele, and in female FOXP3S,
I think they're saying 60% of the females who have this genotype develop tumors by age 2.
oh, okay. And then they say in female fox, okay, the wild type allele is carried by an active X chromosome.
Mhm.
What's an inactivated X chromosome? That's just the mutated one?
Um, so I think that if you it could be something related to like genetic dosage or whatever, like um in order for an XX chromosome pair to have the same genetic material as an XY, one of the X's for the female would be inactivated in order for
Okay.
them to have the same genetic material. Um, and I think that's what the scurfin does.
Mhm. Okay. In female FOXP3 SF plus mice, cancerous mammary epithelium has 8 to 12-fold more mRNA, the transmembrane cell surface receptor ERB2, than does non-cancerous mammary epithelium.
Okay, so they'll probably ask you something about amino acids in the transmembrane domain. Um, but yeah, it seems like the cancerous cells have significantly more ERBB2, and they'll probably tell us what that means or what that does.
Fo- FOXP3 binds specific sequences for the ERBB2 promoter, and deletion of these FOXP3 binding sites increases expression for the ERB2 promoter in FOXP3 expressing cells. Can we draw this out?
Hmm.
Um, so yeah, FOXP3 is a transcription factor. Um, there's an X-linked gene. Wait, it's encoded by an X-linked gene. So yeah, whatever. It binds a promoter, ERBB2. Like, are the binding sites they're somewhere in the promoter, in the ERBB2 promoter.
Let me see. Um, let's see. So I'm looking at that. So the first thing is, or bullet point, even though there's no bullet points, but the first thing seems to say that wild type FOXP3 transcripts, meaning mRNA, are easily detected in non-cancerous mammary epithelium from the FOXP3SF plus mice, but are undetectable in cancerous mammary epithelium from the same mice. Um, and then in female, the wild type FOXP3 allele is on, let's see, on inactivated X chromosome in like 15% of non-cancerous, but in 100% of cancerous mammary epithelial cells. And then we say that um, the ERBB2 um, mRNA is going to be in cancerous, whoops, the cancerous mammary epithelium, like significantly more so. And then we say that FOXP3 binds specific sequences in the ERBB2 promoter, and deletion of these FOXP3 binding sites increases expression from the ERBB2 promoter in FOXP3 expressing cells. So, okay, if we delete the FOXP3 binding sites, we have more expression of ERBB2, which makes me think that if FOXP3 binds to ERBB2, it should cause FOXP, or it should cause a decrease in expression. FOXP3 binding to ERBB2 inversely proportional to expression of the FOXP3. The growth rate of mammary tumor cells in culture and the ability of these cells to cause death when injected into mice correlate inversely with the level of FOXP3 expression, meaning the higher FOXP3 expression, less ability for mammary tumor cells to die. So I say proportional, or rather, cause death. Growth rate of mammary tumor cells and ability of these cells to cause death when injected into mice inversely correlate to the level of FOXP3 expression. So higher FOXP3 expression means lower growth rate of mammary tumor cells and higher ability to cause death. So I would say put a semicolon. FOXP3 expression proportional to inversely proportional to tumor growth, and wait, yeah, and inversely proportional to the apoptosis, the cell death. Wait, growth rate goes down with FOXP3, and apoptosis goes down with more FOXP3, right? So far I see that the amount of wild type FOXP3 transcripts, aka mRNA, is higher in non-cancerous mammary epithelium. Um, it's also higher on the inactivated X chromosome in 15% of non-cancerous mammary epithelium, but it's on that inactivated X chromosome in 100% of cancerous mammary epithelium. And ERBB2 mRNA, meaning transcripts, are going to be more present in cancerous mammary epithelium. Um, and FOXP3 binding to ERBB2 causes less expression of that FOXP3. And the more expression of FOXP3 that you have, the less tumor growth and less apoptosis you'll get as well.
Mhm.
So that's what I take from all of this.
Hold on, I'll be right back again. I'm sorry, my roommate just kind of causing noise.
Yep.
Okay,
Mhm.
all good.
Yeah. Okay.
So,
Yep.
basically, to summarize, like you have the wild type Fox P3 transcript. There's a lot of them. They're easy to detect non-cancer cells.
Hmm.
They're undetectable in cancer cells.
Okay.
Um, and what does an inactivated like X chromosome mean? Like, I don't understand.
Yeah, so I think what that means is um let me see. Like I was thinking that it meant yeah, so it is what I thought. So basically gene dosage it's called. So um okay, so a female with XX chromosomes and a male with XY chromosome. The Y chromosome is really just a kind of like a broken X chromosome. So if you're comparing a male with XY and a female with XX, it would seem that the female would have double the amount of genes from having two X chromosomes compared to the male, which has one X chromosome. Like it would give you the end result of the female having twice the number of genetic material, right?
Right.
So what happens is that the X chromosome in the female can become inactivated so as to have the same number of DNA or, genes or genome as the male would have. So, uh, so from looking it up, it says X chromosome inactivation is the form of dosage compensation in mammalian female cells to balance X-linked gene expression levels of the two sexes.
Mhm. Okay.
So it's something that um, would happen to essentially, you know how like in meiosis for females, you have like one chromosome and the other one is like a bar body,
Mhm.
right?
I forgot what that is though. I haven't heard of that term in such a long time. Or a body. Oh yeah, condensed and active X chromosome.
Mm-hmm.
It's found. Got it. Okay.
Yeah, so one of the X chromosomes for the female will become inactive to turn into like that bar body. So you have the same amount of genetic material in both the male, which has just that X chromosome, and the female, which has two X chromosomes initially, but one of the X chromosomes gets inactivated and turns into a bar body.
Okay.
So they have ultimately the same amount of genetic material.
Hmm.
Mhm.
Okay.
Yep. So that's what the inactivated X chromosome means.
Okay. So 15% non-cancerous, 100% cancerous.
Mhm.
Okay, and the cancerous ones have a lot more mRNA for year B2,
Yep.
but what I just don't understand, it's like I just don't understand. Like if there's a lot more year B2 for the Fox P3, if you delete the Fox P3, there's more ERB?
So there's more ERB transcripts, aka mRNA, in cancerous mammary epithelium. And there's and FOXP3 can bind to the ERBB2, and that can cause um less expression of FOXP3. So, and if you have less expression, that can mean you have um or sorry, FOXP3, if it binds to ERBB2, it causes less expression of FOXP3, and FOXP3 expression can cause um, a decrease in tumor growth and apoptosis. So basically, here we see that cancerous mammary epithelium has a lot more mRNA for ERBB2. Here we see that FOXP3 binds to the ERBB2, and if you delete the binding sites, you get more expression of ERBB2, which means that the binding sites, if you have them, you should have less expression of the of this.
Conceptually, I just don't get how that makes sense, but okay.
Yeah. So sorry, I wrote inversely proportional to expression of Fox P3, but it really means expression of the from the ERBB2 promoter. So I'm going to change that.
Hmm.
Fox P3 can bind to ERBB2, and that would reduce the expression of the ERBB2. And the growth rate of mammary tumor cells is inversely proportional to the amount of FOXP3 expression. So if you have more FOXP3 expression, you have a decrease in the growth rate of tumors, which is what I wrote here. But also the ability of these cells to cause death when injected into mice, apoptosis, is also inversely proportional to the amount of FOXP3 expression. So while FOXP3 expression might reduce the growth rate of mammary tumor cells in culture, um, it would also reduce the ability of these cells to cause death. Um, so it should also cause a decrease in the apoptosis.
Okay. Um, Based on the passage, FOXP3 affects ERBB2 expression, non-cancerous mammary epithelium, most likely by directly. Um,
Okay.
okay. So,
So first, let's see, right? We're talking about A and B are talking about mRNA, C and D are talking about protein, right?
yeah,
Now, when we talk about expression, what are we talking about? What
mRNA.
good. So we should be able to eliminate C and D, right?
Yeah, on the exam I remember. I did that. I eliminated C and D.
Yep.
That I
That
remember.
at the translational level, which is not what we're looking for. We're looking at the transcriptional level. And now we just want to see, okay, does FOXP3 uh cause less ERBB2 to be expressed or more?
Um, More.
Over here?
Wait. Okay. Oh wait, no. Yeah. Not so less.
Yeah, so this is
When
saying
it binds, there's less. Yeah, yeah.
yeah, when it no, so it's saying when it binds, it can so it says okay, so when it says the deletion of the binding sites, meaning when FOXP3 does not bind to ERBB2, right? You would have more expression of the ERBB2, right? Like deletion of the FOXP3 binding sites increases expression of ERBB2, right?
Okay, and that means it's an inhibitor because if it does bind, there's less.
If it binds, there's less, yeah. So it'll be a purport, yeah.
Okay, I actually got this right.
Mhm.
Next two I didn't though.
Okay.
Okay. This one, oh my God.
Mhm.
This one, like, yeah, I was just
All right, let's
okay.
let,
Given that the tumorigenicity of a certain mouse memory tumor cell is dependent on ERBB2 mediated intracellular signaling, mice injected with variants of this cell line that have which of the following modifications would be most likely to longest survive? Most likely to survive the longest.
okay. So we know that FOXP3 is inversely proportional to the like the presence of FOXP3 binding to ERBB2, right? Causes less expression of ERBB2. So if they're saying that the genicity of this tumor cell line is dependent on ERBB2, what would cause, I guess, these cells to survive the longest, aka not have apoptosis happen? So this thing over here is proportional to FOXP3 expression. So we want more FOXP3, right?
Okay. More FOXP3, yeah.
The ability of these mammary tumor cells to cause cell death inversely proportional to the amount of FOXP3 expression, right?
Hmm.
So we want FOXP3 expression to go up, right?
Got it. So
For less apoptosis, yeah.
Okay. So here's the thing. B we can eliminate because we know Fox P3. It has to be Fox P3 here.
So we know Fox P3 would mean less ERB B2 expression, right?
Okay.
From this.
Yes.
So if you have ERB B2 genes amplified, you should have less Fox P3, which is the opposite of what we want, right?
The thing is though, I thought I thought that Fox has a effect on ERB too, but they're not. It's not a reciprocal relationship. Like it doesn't go both directions.
Yeah, so if Fox P3 binds to ERB B2, we have less expression of ERB B2. But also, we know that Fox P3 expression is inversely proportional to tumor growth and inversely proportional to apoptosis. So
Yeah.
they're asking yeah, which modifications would cause the cells to survive the longest. So we want to see um well, let me see. Yeah, so they're asked so surviving the longest would mean an inverse proportion to apoptosis, right?
Yes.
So and that happens when we have an increase in FOXP3 expression, right?
Hmm.
So um so that's what we want to be looking for then, right?
Yeah.
And we also see that FOXP3, like if it binds to ERBB2, it causes less expression of ERBB2. So we want we want either our FOXP3 to amount to increase or our ERBB2 amount to decrease.
Okay, so the one of the main reasons I want to go over this with you as well is just to also go through these different answer choices and
Yep.
why they're wrong. Well,
Mhm.
look, so with A, I also don't know I don't know the right answer, but with A, I would guess that's probably the correct one because my reasoning is if you delete the FOXP3 gene, that would mean that there's no FOXP3 that's being there's no transcription factor FOXP3 that's being expressed if you fully delete the gene. And if there's no FOXP3 expression, then right?
So
Oh wait, no, you want FOXP3 for
yeah,
for survival longs. Oh my God.
this one, right? So
Oh, okay.
let's look at it this way, right? So there's more FOXP3, right, in non-cancerous tissue, right? And that's just in general. Now FOXP3 can also be on an inactivated X chromosome in 15% of non-cancerous tissue, but it's on that inactivated X chromosome in 100% of cancerous tissue. What does that mean? That means that cancer is happening when you have the FOXP3 gene inactivated,
Okay.
right? And then we also are being told that the amount of ERBB2 mRNA is significantly higher in cancerous tissue, and that when FOXP3 binds to ERBB2, it causes less expression of the ERBB2. So if we can see that ERBB2 is proportional to cancer and FOXP3 is inversely proportional to cancer, we can then say that Fox P3 expression is, oh, and then based on this last one here, the Fox P3 expression would cause less of the tumor growth and less of the apoptosis. So for this question, it's asking about which modification would cause the cells to live the longest, aka inverse, like what would cause an inverse proportion to apoptosis. And we know so this inverse proportion of apoptosis is proportional to FOXP3 expression. And we also know, so I could write FOXP3 expression is proportional to, or rather inversely proportional to, let's say, cancer, and also inversely proportional to ERB, ERBB2, which means that we can say ERBB2 is proportional to cancer. So so this question is asking us, what can we, which modification would produce the least amount of apoptosis? And we know that that would mean a high amount of FOXP3 expression or a low amount of ERBB2 expression.
Oh.
Right? So that means what we're looking for is a high amount of FOXP3 or a low amount of ERBB2.
Okay.
So let's see. So A says deleting the FOXP3 gene. So in order for us to produce cells that will survive the longest, aka inversely proportional to apoptosis, we would want increase in FOXP3 expression, right?
Yeah.
So A would be the opposite.
Hmm.
Now, we also know that ERBB2 is proportional to the cancer and inversely proportional to FOXP3. So we want less ERBB2, not more. B says more ERBB2, so that's out. And then C says a FOXP3 expressing plasmid is introduced. And we know FOXP3 expression is inversely proportional to apoptosis, which means directly proportional to survival of the cell. So C would work. And then let's see, D says the FOXP3 binding site's deleted in the promoter of one of the ERBB2 genes. We said that here. We said that if you delete the FOXP3 binding sites, it increases the expression of ERBB2. So D would cause ERBB2 to go up, which we know is what we don't want. So that's out as well.
Okay. I think on the exam I screwed up the proportion, and so that's why I might have been confused because A, B, and D are all aligning with the wrong proportion, which I got wrong. So yeah.
Yeah. So what could have helped is when if you just evaluated the answer choices and saw that okay, is saying um A is saying less, maybe think about it like that, right? A is saying less FOXP3, B is saying more ERBB2, C is saying more FOXP3, D is saying more ERBB2.
Yeah.
So we could then say B and D are both saying more ERBB2, so we could eliminate those. And then between A and C, it's a matter of do we want more FOXP3 or less FOXP3? And then you can attack it that way.
Okay. Yeah. In a female mouse born with two wild type FOXP3 alleles, what is the minimum number of inactivating recessive mutations that might be sufficient in the FOXP3 alleles in mammary epithelium to significantly increase the likelihood that this mouse will develop mammary tumors? Okay. Two wild type FOXP3 alleles. So yeah, for this, I was thinking one
Tell me your reasoning.
because like there was one inactive and one wild type,
Uh-huh.
and 60% of them developed a tumor.
Yeah. Minimum number of inactivating recessive mutations that might be sufficient in the FOXP3 alleles in mammary epithelium to significantly increase the likelihood that this mouse will develop mammary tumors. So, yeah, I guess you just need one inactivating scurfin allele to
What I recall, I think I put one and got it wrong.
so let's see. So this is one scurfin and one wild type.
Yeah.
And this is a female mouse born with two wild type. Minimum number of inactivating recessive mutations sufficient and significantly increase the likelihood that this. Okay, if you have one scurfin allele and one wild type, 60% of your stuff is going to be messed up.
Hmm.
If you have two wild type, minimum number that will be sufficient. Okay, you know what? I think that this inactivated scurfin allele is what causes that X inactivation. So if it causes the X inactivation and you have two wild type, we're inactivating one of them, right? Then I think you'll only need one then. Because if
Uh,
you have just one, that'll cause that like one of the X chromosomes to get inactivated.
Got it. Okay, sorry. That's my apologies. I put two. So I guess what because this is the last passage I did, I'm pretty sure, or pre-last. And so yeah, I remember thinking that oh, XX, you need two of them to be recessive
Mhm.
for it to show, you know? But yeah, yeah, exactly. It activates single activating allele. Yeah, I mean, the passage told us this information.
Yeah, but the passage said that for one wild type. So I mean, logically, I could see two being an answer choice. I think the only thing that led me away from that is to think what does it mean to be inactivating, and that means you're inactivating the X chromosome. So if you have two alleles and you inactivate one anyway, it doesn't matter if you have two wild type X whatever, two wild type alleles. Because all you need, if you just inactivate one, if you have one of the scurfin things, you inactivate one of the X chromosomes anyway. So it doesn't matter if you have two to begin with or one. But I think that that's tough. I probably would have chose two as well.
Okay. That's actually it. I mean, I this one I understood. I got it right.
Okay.
So, yeah.
Yeah, this is a tougher passage type. that I think I usually notice towards the end, where it's like no charts or anything, but it's kind of like a logical, like a statement of
Yeah.
false things that are just, if you can make like a logic kind of table based on that. Um, and also like it's worded in ways that are very confusing. Like, for instance, saying, uh, your BB2, Fox B3 or whatever defines that you're BB2. And if you delete those binding sites, you get an increase in your